effect of that. So you might say, so what? If this is converted to this one, what happens? So this is more stable. It's not digested by the enzymes. It is accumulated in neurons and destroy the nervous system. I'm not explaining these ones, but let's go to the two examples of proteins. Then you have one protein with two examples of what? Can you complete the sentence? I'm saying that Tauna alakber. What does it mean? So what's the rest? What it says? It says work together, help together. So hemoglobin is the best example of Tauna alakber. I'm going to explain why. Myoglobin is like this, just one protein, one polypeptide. It's a polypeptide. It is folded correctly in myoglobin. Where do we have our myoglobin? In muscles. In many places, but specifically muscles. What about the hemoglobin? Hemoglobin isn't one polypeptide. It has four of them. 1, 2, 3, 4. Four of these peptides group together. It seems like 4 friends. So, let me ask you a question. What is the size of the red blood cells? Can you look for that, search for that? Size of the one red blood cell. The size of red blood cell, RBC. The size of RBC. What's that?
7 to 8 micrometers.
Micrometer. Micrometer. Millions of meter. So, yes, inside the red blood cell, what we have? Hemoglobin. Red blood cells transfer oxygen. So, with what? With hemoglobin. How many of hemoglobin we have that on that size? In just one red blood cell. 6 micrometers, 6 to 7, 8 micrometers. So, micrometers, millions of the meter, a meter, millions of that, 6 micrometers. How many of hemoglobin we have inside one red blood cell? What do you guess? How many? Just make a guess.
2 or 3.
2 or 3. Higher. It's an option. 5. Go on. How many hemoglobin inside the one red blood cells?
What? 10?
Go on.
100?
Go up.
1,000.
1,000, go up.
1 million.
1 million, go up.
100 million.
Go up.
1 million.
250 millions of hemoglobin inside one red blood cell. Do you believe that? Do you believe that?
That's possible.
It is possible because the hemoglobin is so small, nanometer. And we have 200 million, more than 250 millions of the hemoglobin one side red blood cells. That is a real magic. And soccer? So, how many of oxygens transferred by one red blood cell? Each hemoglobin is hemoglobin. Each hemoglobin, how many of units we have in hemoglobin? How about the myoglobin? We had just one O2 for myoglobin. For hemoglobin, it has 4 subunits, 4 units, 4 O2. So one red blood cells can carry or transfer how many of oxygen? 250 millions multiplied by 4. 1 billion of O2 transferred just with one red blood cell. One small red blood cell. So you are going to see that tau monoallel pair with these 4 friends in hemoglobin structure. They work together and release the oxygen with helping together. So, do you have some of those, heating system? They release CO, they consume gas. What kind of heating system do you have in your homes? What's the heating system in the winter? Do you consume gas? So what's the side product of that? There is always toxicity for gases. What's that? Carbon monoxide. Why carbon monoxide is toxic? It is a killing agent. It is called silent killer because without any signs, it can kill everyone. Because it has affinity or love for what? For hemoglobin. CO is, kind of compound, likes to bind to hemoglobin. So oxygen and carbon monoxide, both of them like to bind to hemoglobin. But what is the difference? Which one likes more? CO or carbon monoxide likes 200 times more than oxygen. So because of that, it can bond so easily to hemoglobin. When it bonds to hemoglobin, it doesn't let release the oxygen or transfer the oxygen. And because of that, intoxicated people, they will die in high concentration of CO. Because CO, see, this is a hemoglobin structure. Let's say one of these. We had 4 of them. Hemoglobin protein, when I talk about the hemoglobin, I mean these 4 proteins together. That is called hemoglobin, one hemoglobin. How many of oxygen transfer? 4. 1, 2, 3, 4. Inside each of them, we have a capacity. This is a globulin part, protein part, and the other part is heme. Inside the heme, what we have? What is the main, actually, one of those compounds for anemia or blood making? Is iron. Some people, they face with iron deficiency, specifically women, they need iron supplements. Because iron is a main part of this hemoglobin part and myoglobin part also. So it bonds to the oxygen, this iron, and transfer this oxygen in blood circulation. Instead of this O2 or oxygen, CO can bound to this here. So because of that, CO is toxic. Why low concentration of CO is toxic? Because CO likes more than O2 here. It bonds so tight and you can't actually release it so easily. So because of that, CO is very toxic compound. So this is the O2 bonds to the iron. This is the heme structure, like this hemoglobin part. We have iron on it, and it bonds to O2, and also it bonds to CO. So it is better to protect yourself from the places that they have a lot of CO, because CO is a killer agent. And what is the role of the hemoglobin? Transfer O2 to what? To tissues. So what was the product of the tissues? ATP, we burn that carbohydrate, we produce what? ATP for energy, CO2 and water. So see what happens to that CO2?
Exhale.
We exhale. How we transfer from tissues to lungs? By hemoglobin. Hemoglobin transfer that CO2 to the lungs, and the lungs actually help to get rid of that compound. So it is the role of that. So in the tissues we produce CO2. CO2 is transferred by hemoglobin to the lungs, but in three forms. All CO2 that we produce in the tissues are transferred in three formats. Some of them are dissolved in water or blood. What does it mean? Directly, like soda, carbonated soda, when you open that, it has a gases on it. If you look at our blood, we have a 8 to 10% gas on it. That gas is CO2. But we can actually have more than that. How about the rest of that? That's the 10% of CO2. The rest, 70% of that, or 60 to 70%, is bicarbonate, is a buffer. What is the buffer? When you drink acids or bases, what happens? Your body can control that pH changes with this one. This is a buffer system. And some others bound to the hemoglobin. The rest are bound to the hemoglobin. So see, what is the idea? You have a protein, gets oxygen from lungs. What happens to that oxygen? Goes to the tissues, all tissues. They consume that O2 to produce what? CO2. What happens to that CO2? Part of that is dissolved in the plasma or blood. Part is bound to the hemoglobin. Main parts of that converted to other chemical compounds. What is that chemical compounds? Bicarbonate. What is that? It helps you protect from pH changes. When you consume acidic or basic compounds, this is there. Help you to control acids and bases. It doesn't let changes so much. So this is the role of the hemoglobin. But also there are some parts in this hemoglobin structure bonds to hydrogen. Hydrogen is acid. When your tissues produce acids, when you do some activity, produces acids. But as that acid bonds to the hemoglobin and helps to neutralize that. So there are some questions around this. So these 4 units, see, hemoglobin has 4. Do you remember that 4?
Yeah.
2 of them alpha, 2 of them are called beta. The names of the proteins. Alpha is alpha peptide, beta peptide. Don't remember those things. Just know that we have this kind of thing. But there is another magic here happens. What's that? If you go mountains, what happens? The concentration of O2 is low. Your hemoglobin should release oxygen so easily because in that places you don't have enough oxygen. Your hemoglobin should release that one easily. How? With the compounds. That red blood cell produce that one. It is called 2,3-diphosphoglycerate. Do I have full name here? Yes. 2 and 3 bis or di. Both of them are the same name. This bis or di, they are same. Bisphosphoglycerate is a compound goes inside this 4 protein and force them to release the oxygen. Why? because you are going to places the oxygen level is low and your hemoglobin should release that one. That is another magic. And remember these names just as a different name, alpha and beta. Let's say normal hemoglobin in adults. But, great question. How moms transfer oxygen to the fetus? Have you ever think about that?
Why the placenta?
You don't have mixing blood. There's a barrier there. Just oxygen transfer to the other side. How it happens? If they have same hemoglobin, they don't have same hemoglobin. For fetus, we have hemoglobin F or fetus hemoglobin. That was adult hemoglobin. So what is the difference between fetus and adult hemoglobin? There are some small changes on hemoglobin. What is that? That hemoglobin likes more than adult hemoglobin for oxygen. It likes more. It can get it easily. And moms can release oxygen so easily. Oxygen released to the blood and the other side, hemoglobin can get that like lungs. It happens in the lung. So, because of that, we have different types of the hemoglobin.
I don't understand the process of fetus and blood.
You have a barrier, not mixing mother's blood with fetus.
So the blood will, the oxygen will transfer to which structure between fetus and
So you can have 3% to 5% of oxygen dissolved in water. You can have that. So from when actually fetus blood is around that, it can get easily that oxygen. You will have a specific concentration direction from mothers to the fetus. So that 3% goes there, that hemoglobin gets that, brings it to the other tissues, and another circulating hemoglobin comes there and get that oxygen. That is different types of their liking is different for the hemoglobin. That hemoglobin, there is a P50, let me explain on the next slide. What is this? What does this say? You know, this is slide. It says, see, this is a PO2 pressure. But in what pressure, what percentage of the oxygen are saturated? For example, you have 250 millions of the hemoglobin in one red blood cell. How much of them or what percentage of them are fully saturated? How much of that? It depends on the oxygen pressure. So, 100%. In what pressure we have 100% of saturation?
100.
Around the 100 milligram of this mercury. But how about the half of that? In adults, it is 26. In 26 millimeter of this pressure, because in many places we don't have this pressure. In activity, what is the oxygen pressure? It is low. In mountains, it is low. Some people, they have diseases on their lungs. The O2 pressure isn't 100. This is a perfect one. So in many cases, it is lower than that. So our body has a capacity, see, in 26 milliliter, half of that oxygen is full. It has, let's say we have a 10 hemoglobin here, and in 26 millimeter of this oxygen pressure, half of them, or 5 of them, they have oxygen, full oxygen. How about the kids or fetus ones? It is 20. In 20 millimeter compared, you have a group here, on 26 millimeter of the oxygen, they are fully charged, The others, that is the fetus hemoglobin. In what pressure they are put? 20 milliliter of this pressure. So low pressure, they can get oxygen from the lungs so easily because of the some differences that they have, and because of that we have different types of the hemoglobin. See, these are different types in different stages. For example, this is for adults. This is minor adults form. This is fetal one. These are in embryonic states. Different hemoglobin in embryonic states. And diseases, 2 diseases related to hemoglobin. 2 diseases. Again, you know the normal ones, and you will see some diseases related to that. What are the diseases here? If this one is a sequence of one peptide, we have amino acids on this structure. Do you remember that?
Yes. For that
Yeah.
correctly, we have skeleton.
We have amino acids on this structure, normal ones. Imagine one of these amino acids changes. It will not fold correctly. It will be like this, for example. Let's say it doesn't let it fold correctly. So, what happens? This protein will not work properly. It happens for hemoglobin. Just look at this part. What is the difference between these two? What happens here? What do you see here? Just changing in DNA structure, thymine to what? Adenine. What is the result? Changing these amino acids, normal one. Glutamic acid to what? Valine. What is the effect of that? Cause diseases is called sickle cell anemia. Just one nucleotide. See, in DNA, thymine converted to adenine. Results changing this amino acids because this code GTG is for glutamate, but GAG is for valine. And the effect is unbelievable. Instead of this
Complete circle, we have
these red blood cells should be so flexible. Instead of, you will have some red blood cells like this, like cycle cell. They have a bend on that. So a lot of these things can trap together. When you have a lot of these things, they can actually clog together in small vessels and make a trap there. So what happens? What happens? This is the shape of the cells, but what is the result? It's a kind of hypoxia. They can transfer oxygen, and they don't let oxygen go to the tissues. What happens to the tissues? Necrosis, or dying the cells. So just one nucleotide changes. One nucleotide changes amino acids. Final effect is that you might say, how many of people we have? They have this disease. We have a lot in Africa and in Middle East. Sickle cell anemia is one of the most common diseases related to hemoglobin.
I read in one time that sickle cell anemia is separated near the regions that near the sea.
Yeah,
What's the relation?
yeah. I'm going to explain that. Why? So, another one is related to not changing the structure. In previous one, in sickle cell, we saw it was just one changes in sequence of that. But in some diseases, we have one of these alpha or beta are not synthesized. We don't have one of these strands or pair of them because they were as a pair, 2, alpha 1, alpha 2, beta 1, beta 2. Another disease is called thalassemia, is the name of that. if you look at the name of that, etymology of thalassemia, you can look for that. Just look for that in your internet. What is the thalassemia means? I need someone to explain that. What is the mia? Emia?
Blood.
Blood, exactly. Or low level of blood.
It's groups of inherited blood.
Yeah, they are inherited diseases, but what is the thalassemia? You mentioned about the a specific definition. I'm just trying to show you what is the meaning of that. What does the thalassemia mean?
Manifest of protection of reduced hemoglobin.
You are correct. You are explaining that, but I'm going to
Thalassemia means C.
C. Again. So explain that. C.
Yes, anemia, blood.
Blood. Diseases for blood around the sea. Because for first time it was diagnosed around the Mediterranean Sea. So we had this science around the Mediterranean Sea, in Egypt, in Greece, or in Middle East area. So because of that, for first time, the name comes from that. The disease was around the Mediterranean Sea.
So
Thalassemia.
what is the relation? Why when I don't understand the relation? Why when they live behind the sea they
So you might say, it comes from the region, or mainly it is related to some marriages happens between this group that they have these diseases. Yeah, it happens because we have this kind of diseases in West country and North America. People that you might see there, they are from the Central Africa or Asia. It is more specifically on this area. It mainly comes from the marriages happens 2,000 or 1,000 years ago. We don't, some, inter-tribe marriages, they actually transfer this kind of diseases. So, what is the thalassemia? If you compare with the sickle cell anemia, in sickle cell, we had just small changes on this structure, but in thalassemia, we don't have alpha or beta parts. Because of that, it is called alpha thalassemia or beta thalassemia. It is related to low level of the hemoglobin. We can't synthesize enough hemoglobin. In sickle cell, we synthesize that, but it's not applicable. We can't use it. It has a problem. Here, we don't have genes or some genetic genes. I guess in your country, do you have screening before marriage for, thalassemia? Because thalassemia is more prevalent in Middle East. Yeah. It is related to not having one of these genes. For the beta, we need two genes, one from father, another from mother. In cases, just we miss one of them, for either father or mother. If we have one of them, we call it minor thalassemia. But if you receive, the malfunction or not having beta from both sides, what is called major thalassemia. It is a kind of more severe one. In minor one, you don't show, in beta thalassemia. In minor one, you don't show signs of that. But for the marriage, two minor, they have a risk, have a 25% having a child, what? With major thalassemia. For their actually kids, they might have minors, for all of them. There is a chance for that. But also chance for 25% of, if men and women, both, they be minor. How about the alpha thalassemia? It's the same to that, but is less complicated. Because for the alpha thalassemia, I'll be back. For alpha thalassemia, we have two genes from mom, two genes from father. So the probability of the disease is less. Because in beta, we have one gene. Just one gene from father, one gene from father, but for the alpha, we have two genes from father, here, two genes from father, two genes from mothers. And also, in cases we don't have whole of this gene, see, it says not having. They are empty or we don't have alpha. This is a kind of hemoglobin parts. They will die at birth. Or this one is a hemoglobin H disease. They show severe. They have just one alpha, they don't have other ones. And so it seems the hemoglobin don't have alphas, just betas. And it doesn't work properly. I said they work as a tau alpha. Without the other parts, they will not work. Why I use that phrase for that? Because they work together. Without friends, without their couples, they can't work. So, yeah, this is the hemoglobin part. So, yeah. If you have any question, sorry for that.
In psychoanemia, Dr. you mentioned this disease happened because change in nucleotide.
Yeah, nucleotide.
So my question is, what's the cause of these changes in the gene?
Oh, yeah. You know, most of the time, you know, when we replicate DNA, there is a chance to mistake, always. But our body have proofreading. Read that, make that, you know, mistakes correctly, and yeah, solve the problem. But in some cases it happens, we don't know. Some chemical, some reading area, living area, like, you know, epigenetic effects. We call it epigenetic effects. If you live in an area faced with some specific foods, specific diets, I mean, not normal diets. Some people actually get alcohol or some, you know, drugs or some medication. In some generation, they might have effect on their genes, and it is called epigenetics. And so it could happen. But if someone have genetically that one, we have a genetic subjects. Some of these genes are, you know, transferred to next generation. We know the science. There are some other mutations on DNA, but their effect isn't so severe. This one is, we have more than 1,000 mutations in DNA, in hemoglobin. But the more severe one is sickle cell anemia. However, we have in non-protein parts, promoter parts. They actually silent the genes, like beta thalassemia. You don't have genes.
So this disease could be a genetic or hereditary disease?
Yeah, they are hereditary disease. So, yeah, that's it for this part.
I got broads in Atlanta, took the lean in the family, credit cards in the stomach, it's in the back.
Why is your family dying? You need some?
You know what's this? Do you remember this animation? What's that? Exactly. Who likes that? Yes. So let's have 5 minutes of this. And after this, I want to explain what is the link between this and next session. If you have any questions, just ask me. But it is enough.
So we are really fighting hot and every minute counts. Wait, they're all sloths? You said this was going to be quick.
I thought Zootopia anyone could be anything. Flash, flash, 100-yard dash. Buddy, it's nice to see you.
Nice to see you, too.
Hey, Flash, I'd love you to meet my friend. Darling, I forgot your name.
Officer Judy House, CPD, how are you?
I am doing just
Fine?
as well as I can be. What
Hang in there.
can I do
Well, I was hoping you could run a play.
for you
Well, I was hoping you could.
today?
Well, I was hoping you could run a play for us. We are in a really big hurry.
Sure. What's the plate
29T.
number?
29THD03.
Two, 9,
THD03.
T,
HD03.
H,
D03.
D,
03.
0.
3.
Hey, Flash, wanna hear a joke?
No!
Sure.
What do you call a three-humped camel?
I don't know. What do you call a
3-pound camel.
3-humped camel?
Pregnant.
Yes, very funny, very funny. Could we please just focus on the text?
Hey.
Wait.
Priscilla,
Oh, no! Yes? Flash?
what do
No!
you call
A 3-humped camel? Pregnant! Okay,
Three.
great, got it. Please just...
humped? Here
Yeah.
you go.
It's registered to Tundertown Limo Service. A limo to Robertson and a limo to Tundertown. It's in Tundertown!
Way to hustle, bud. I love you. I owe you.
Hurry! We gotta get fresh hour in. It's night!
So, what's the link between this and enzymes? Can you make a guess? What are the enzymes? What they do? What the slants are so slow? Why?
The enzyme can control the
Yeah, kinda.
formation.
Yeah.
The enzyme analyzes the food into small pieces, like the protein. When we mentioned that in the antibody, the protein is divided or analyzes to small pieces, which is amino acids.
Yeah, you are right. But the main part is that for enzymes, actually, what are the enzymes? Enzymes are biological catalysts. What are the catalysts? They increase the speed of everything. See, the point was that these cells, they can't actually work so fast. Why? Their metabolic is so slow. Their enzyme activity or converting something. What is the enzyme? Converting something to other things. Or helping for doing something. So we are going to see what are the enzymes. So, enzymes. I can't see whole names of the sessions here. So, protein and amino acids, the seeds of sugar, I guess this one. So, give me some examples of enzyme that you know. Yeah, pepsin enzyme. And some other enzymes. You know them?
Lipase, amylase, lactase.
Yeah, exactly. Many of enzymes. In digestive system, specifically. Even in the market, do you have some detergent that they have enzymes? Have you ever seen those kind of advertisements? They say we have some detergents, they have enzyme, they help you killing the messes on your clothes. Yeah, you might have in your country. They are enzymes, they help you to digest lipids or some other things on your clothes. Yeah. And in industry, have you ever taken something to help you to digest something? Some people, they have a problem in digestive system. They can't digest some of these foods, and they receive some drugs. They have enzymes. What kind of foods you are recommended after a meal? Do you have something in your country? They say eat this food after the fruit or something like that. After that meal, it helps you to digest that easily.
Kind of soda.
No, they are not good.
Really?
Yeah.
They always advise to stay.
That's not good. About the fruits, what kind of fruits? Have you ever kiwi, orange? Orange is good for vitamin C, for absorbing iron, because it helps for reducing that. How about the others? Kiwi, banana. Banana is not bad, but related to enzymes. Banana has amylase. What happens to banana after staying for a long time in outside? Yeah, how does it taste? It tastes so sweet. What happens? Amylase works there. Amylase removes that glucose from amylose. Do you remember amylose? In the starch. In the starch, we had amylose. In banana, we have amylase enzyme on it. It digests that. What happens to kiwi if it stays for a long time outside? For the kiwi, what happens? It's kind of soft. Why? It has enzymes on it. What kind of enzymes? Kiwi is good after a meal that you eat a lot of meat. If you get some slices of kiwi, it helps for digesting that because it has digesting enzymes. If you put that kiwi on your meat, it can digest your meat in 20 minutes. Yeah, in some places like Iran, they like kebab, they prepare it in one night before, But in some cases, when they are in a rush and they don't have time for putting that, just 20 minutes before the kebab, they start squeezing some of this kiwi juice on it. But if you put it more than 20 minutes, it can digest all meat because it has a lot of enzyme. If you look at that, it even digests themselves. What happens to kiwi after some days? You see that it's softened. It doesn't, because that enzyme works there, digests the kiwi. Even in coconut, we have a specific enzyme. So, about the clinical importance, some of these enzymes are important. If you look at, have you tried some medical laboratory results in your home? Do you have laboratory results for your families at home? Do you have laboratory results for your family at home? If you have, next time try to find some sugar tests, lipid tests, and some enzymes. That enzymes are important in clinics. Sometimes physician, you might be at that point, you look at the enzyme level. Some liver enzymes helps to give enough information about the liver, because the concentration goes up. Or after heart attack, some enzymes goes to the blood, so it is a clinical usage of that. And the best part, using enzyme for skincare. It happens a lot. Some of these, creams, they have some extractions of these fruits. They have a specific enzyme on it. Who is interested in making jelly? Have you ever tried that and putting some kiwi on it? So, does it work? When you put kiwi on it? Why? Kiwi digests that one. These jellies are gelatin, amino acids, proteins, and it digests that. How can you do it? Put your kiwi on freezer, kill the enzymes, or boil the kiwi and then put on it. The point. Yeah, because that's the enzyme. Enzymes are protein. If you freeze them or boil them, that proteins die. The activities die. Like this, they cut these amino acids and proteins. So, you are going to study this. Is it good?
Yeah, it's very easy.
Yeah. No, I'm kidding. I'm not going, I'm joking. We're not going to study this. But, imagine a lot of these reactions happens in one cell. Just one cell. See? Guys, see, all of these reactions happens in one cell, and we don't know. They are doing their actually job, and we don't care about those things. All of those reactions happens by enzymes. Without enzymes, we face with the diseases, a lot of diseases. Some of them are congenital, some of them are related to vitamins, because this enzyme needs vitamins for working. But all of these reactions happens by 6-group enzyme. 6-group enzyme. We are going to just learn the names of those things. 6 group of enzymes catalyze all of this reaction in the cells. In biochemistry, we have to know all of these things. And as people, they want to work in metabolism, because all diseases are related to metabolism. You eat a lot of something, an accumulation of something, or if you have a disease in one pathway, what happens? You will have accumulation of something. These are objectives for this part. We are going to learn the enzyme, what are the enzymes, what are their roles, and what are their classification, and in which condition they work properly. Definition of the enzyme. What are the enzymes?
Proteins.
They are proteins, yeah. What they do? What do they
Freeze the reaction.
catalyze the speed of the reaction? Imagine without the enzyme, you should stay there like this lasts forever. For more than 10 years, one reaction happens. One glucose produces ATP. It doesn't produce that without the enzymes. So living without the enzyme is not imaginable. They are not altered or consumed during the reactions. They are intact. They can do a lot of things. Like, let's say I'm an enzyme here. My name is transferase. Transfer or putting this one from this side to the other side. This is called transfer. I'm not consuming it. I'm just doing that one. I'm not altered during this process. And reusable. I'm here to transfer that, and again, if you have a lot of phones, I'm reusable. So, this is an enzyme, and this is a coenzyme part. You remember that coenzyme I mentioned about? Mainly, these coenzyme plus this protein part of the enzyme, this is a protein part, this is the non-protein part of that. If you remember that vitamin, they were not proteins. They are different compounds. But proteins always are protein. This part, without coenzyme part, is called apoenzyme. Plus this coenzyme is called holo. What does it mean, holo? Holo means total, complete enzyme. This is a coenzyme part here. This is an apoenzyme, and we call it holoenzyme. Mainly, you are not going to say holoenzyme. When you say enzyme, you mean enzyme plus coenzyme. Mainly, these coenzymes are vitamins. And in some cases, some metal ions, like iron, like copper, like zinc. We call it trace elements. Because they are in small amount, that is enough for working. That is enough for working of this enzyme. So, what are the cofactors? Cofactors are non-protein parts of the enzyme. They actually bound to the enzyme, and they are required and essential for these reactions. Type of the cofactors, if you remember, some of them are vitamins. See examples. Some of them are vitamins. Some others are metal ions, like zinc, copper, iron. And sometimes they need some other cations, like calcium, magnesium. They need for working that. Without that magnesium, they will not work properly. So because of that, those parts are essential for that enzymes. About the structure of the enzymes, how it works. Enzymes has a specific part, which is called the active site. Like, if this be a cutter, and it is supposed to cut this one, this part is active site. I can't actually cut this part by the other side. So the part of the enzyme, reaction happens there, it is called active site. See, this is the enzyme. This is the active site. What is this? Substrate. This enzyme. This is the substrate. If I cut this one, the substrate converted to product. So we have substrate, enzyme, the other side, enzyme, product. So enzyme, active site, substrate. Bonds to that, we will have, for example, here two products. Sometimes we have an enzyme, it's going to connect these two together. We have here, connect two, and the other side, our product is larger molecules, just one molecule. Combining two molecules together, it depends on the enzyme. Some enzymes are synthetic, some enzymes are catalytic enzymes. It depends on the pathway. We have two models for enzymes. Two models. Remember active site? We have active site. Some of these some of these active sites, let's say these ones, they are exactly same to the substrate. They are exactly same to substrate. Like this key we have here, lock and key. They are exactly fit together. For this model, we call it lock and key model. The active site of the enzyme is exactly same to the substrate. Substrate sit there and works a reaction. What is the limitation? What are the limitations for these ones? Lock and key. What do you think about that? We have a specific active site, a specific substrate can bind to that. Is it good? Yeah, it is more specific. But what is the limitation? It is just for that one. It doesn't work with the other substrate. Do you like have a specific enzyme, its active site fit to that substrate? Depends on the substrate shape or different types of the substrate, it can fit that one. What do we call it? Induced fit model. Let's say this is an enzyme and I have a substrate like this. My enzyme can change the shape of the active site based on this substrate. For example, sometimes it's like this, sometimes like this. This active site shape can change based on this substrate. So, what we call it? The shape induced this model. We call it induced fit model. For induced fit model enzyme, they can work in a different group of compounds. But for the lock and key model, they just specifically work. Which one of them is good?
First one.
Both of them. Depends on your need. Sometimes you have a specific enzyme works on just glucose. Is it good? Yeah, it's good. Because you think at that time you are just going to work on glucose. Sometimes you say, I want to work on all sugars that they have 6 carbons. Not just glucose. For the glucose, you call it glucokinase, but for the 6 carbons, you call it hexokinase. What is the hexo? 6. Kinase is an enzyme for adding phosphate to that. So you say, yeah, it's good. If I have that option, it gives me more flexibility to work on different compounds. So, because of that, we have 2 models. Some enzymes are more specific, some enzymes are fit-induced model. So, both of them are work. And examples. The best example that I mentioned that. Some of them are more specific, like glucokinase or urease. What happens on urease reaction? Enzyme works on just urea. Let's find another example. Group of the compound. Group of the compound, like hexokinase. Hexokinase, all 6 carbons are done with reaction. How about the peptide bonds? We have a peptidase. It works on all peptides. Digest. If you consume peptide, meat is a peptide. Our enzyme can digest that one. So, based on that, this is a lock and key model. You can read it. I explained all of these things. What is that? This is a specific active site. This is fit to this model. If you compare this compound, is not fit. But this one is more specific for that. How about the lock and? This is the lock and key model. This is the active site. This is the substrate specific to that. What about the fit model? The active site is like this, but substrate shape is like this. You have a diamond here, but the active site shape is a diamond. But this active site changes shape and fit to that substrate. So, based on the specificity, this term is the most important term is specificity. Or in what level enzymes are specific. a single substrate. Example, glucokinase, urea. Then you have a specific names, it says it just works on it. How about this? A group of similar substrate. Similar substrate, like hexose, all 6 carbons. Sugar, they have 6 carbons. Glucokinase, just glucose. In hexokinase, it means glucose, galactose, mannose, fructose, all of them, they have 6 carbons. And it works on them. And a particular type of bonds. What is the example for that? Do you know that? Bonds. What kind of bonds do you know? Between amino acids, between carbohydrates. Between carbohydrates is called glycosyl. Glycosyl. Glyco means sugar. Bonds between them is called glycosyl. What is the enzyme for that? Glycosidase. Glycosidase. The name of for the enzyme. The general name is that amylase. Amylase digests carbohydrates. All carbohydrates, polypeptides. How about another example for the proteins? Peptidase. All peptidase. They digest all peptides. They are specific for type of bond. You have a peptide bond, you have a peptidase. You have a carbohydrate bond, you have an amylase to digest that bonds. These are examples I explained in the previous ones. You can find here. So, what is the role of enzyme?
Increasing the metabolic reaction.
What?
Increase the speed of the metabolic reaction.
Yes. It reduces the energy. Imagine without the enzyme. It shows not human does that. Imagine without the enzyme, we are going to see the enzyme reaction. It seems you need a lot of energy to do that reaction. And or time. It takes a lot of time to happen. But with the enzyme, what happens, it seems less energy for that because enzymes try to do that one. So, just in one sentence, if I explain what does enzyme do, they reduce activation energy. They reduce activation of energy to catalyzing a reaction. Last part of today's is those that said we remember all reactions. We have 6 types of the enzymes. What are the 6 types of enzymes? For some enzymes, for naming them, we have some traditional names for them. For example, for the sucrose, we add sucro. What is this?
ACE.
Exactly. This ACE is an enzyme. For many things, for many enzymes, we have like this. Glucokinase, urease. It actually is related to substrate mainly. Substrate, it works on it, the reaction does on it, and then ACE. Glucokinase, kinase is a kind of aminophosphate. Glucose is a substrate, adding the phosphate, and ACE is an enzyme. Some others are classified or named based on the type of the reaction. For example, what does do this oxidase? What do you think?
Oxidation.
They oxidize. How about this reductase? Do we have reductase here? So how about the reductase? They reduce the reactions. They remove oxygen from it instead of adding that. Some others actually they common names actually for the digesting system like pepsin and trypsin. It depends on the position that they go for digesting that. They have historical names. The classification. You need to learn these 6 names. What do you think about them? Can you make a guess about them? Look at them. What kind of reaction do you see in these groups?
Oxidation and reduction.
Sometimes oxidation, it depends on the enzyme. Sometimes reduction. They reduce or oxidize something. How about this one?
Transfer the energy.
For example, they transfer, this is a 2 substrate, enzyme transfer this part from this molecule to this one. At this part you will have this one without that group, and you will have that group on it, transferase. Transferase are most important because non-essential amino acids synthesize from essential amino acids. They help transfer amine groups to carbohydrates, transferase, aminotransferases. They transfer amine groups to carbohydrate to sugars and make amino acids. How about this? Add water, break it down. Like carbohydrates or peptides, when you consume, you have to add water to that and break it down to units. How about the lyases? I will ask at the end. Don't make a stop here. What's the isomerases? Do you know examples for them? Isomers. What are the isomers?
I know the meaning of isomers. It's similar to something.
Yeah, their formula is similar in writing form, but in the space, their positions are different, right? Like this one. When I'm like this, or I'm like this. They are isomers. Yeah, this is the examples of the isomers. The best example, E and L, they are isomers. Glucose to galactose, they are isomers. Fructose to galactose, fructose to glucose, they are isomers. What is the usage of that? Imagine one mom wants to synthesize lactose. So what was the units of lactose? Do you remember that? Glucose and galactose. Glucose and galactose for lactose. So these units, if you don't consume galactose, what happens? Mom, they can't actually synthesize lactose? No, they can. They convert glucose to galactose, they themselves, by this enzyme, isomerase. If you consume galactose, what happens? You convert galactose to glucose in body with isomerase. We have a specific pathway for glucose. So if you consume galactose, you don't have a specific pathway for that. You have some part, but mainly they are converted to glucose and glucose then to fructose. We have a specific pathway for that. So this conversions is a kind of done with isomerase. How about the ligases? Sometimes you need to connect more groups to this ones. Let's say, not transferring. I'm gonna make a fuse of this. Add to this ones with energy. The only one that needs energy for reaction is this one. The rest, they don't need energy. See? They don't use ATP. They might transfer ATP or putting money on pocket, but they don't consume that. Just except this one. This one consumes energy. So adding a group or fusing something to something. So, how about these ones? Liases. This is a kind of complicated one between them. Among these groups, this one is a kind of more complicated because sometimes it adds water, remove water, making new compounds. It's a kind of enzyme. The difference between these hydrolases and lyases, what was the main definition for hydrolases? You add water, break something to units, not breaking it down. let's say if I have a lot of sugars here, units, adding water remove the sugars, units of this monosaccharides.
Breaking the chemical bond.
Exactly. It breaks in hydrolysis, you add the water to release units, just small units of that. But in lyasis, you destroy that. That units destroyed, not large molecules, polymers. That the difference is that. So you are going to just learn the name of this. That's enough. On next slide, you will see examples, all of those explanations. What is this? What do you think here? Examples. Let's try your science on this part. Oxidation and reduction. What is the dehydrogenase? It's a kind of oxidation and reduction. What is this? Transaminase. It's a kind of transferase. What is this? Adding water to breaking this one. It's called hydrolysis. That's so easy. This is the lyases, add water, but changing these molecules, not removing units I mentioned. See? Changing that, rearranging that, not removing part of that. What is this isomerases? You converted L-alanine to what? D-alanine. And this is the isomer for glucose to fructose. Converted that. And finally, you have a molecule here. You want to add to this amine group to this new bonds with consuming what? Just one group consuming ATP. The ligase. All of these two slides, next two slides are explanation of those ones. Everything that I explained you can find here. And these two. So, the last parts of the enzymes are environments that affect enzyme activity. One of them was cofactors. In deficiency of vitamins, enzyme will not work. So you should get enough vitamins. How about the unenvironmental? Environmental condition is related to pH and temperature. In high temperature, enzymes are protein, they will die. Because of that, we can tolerate more than 45 temperature. In 42, that is so high fever, and we can tolerate that. In 45, we will die, because many of these proteins will die. And enzyme inhibitor. The most interesting part is that controlling enzyme activity. With what? Drugs. Many of drugs or toxins, they control these enzymes. They are called enzyme inhibitors. They are not necessarily bad things. Sometimes we want to control our enzyme activity with drugs, like anti-blood pressure drugs or painkillers. They actually stop enzyme activity. Some toxins also kill the enzyme. Or antibiotics. Antibiotics stop bacterial enzymatic system for synthesizing membrane or synthesizing proteins. So studying the enzyme is the most interesting subject because all reactions happen with enzyme and you can control them with inhibiting them, targeting them a specific drug. I'm not going to explain one by one these ones. I explained this environmental for the temperature. See, the optimum temperature is around 37. In low, this is the turnover number or activity of enzymes. It is low when the temperature is low. When the temperature goes up, it is going up, and the optimum is here. If you increase the temperature more than 40, 45, see, in this part, you don't have enzyme activity. This is the enzyme activity, it goes down. And also about the pH. Some enzymes work on acidic pH, like pepsin in our stomach. Works on this pH. If the pH changes, it doesn't work. It doesn't digest because it works on this pH. And for the amylase, it works on a neutral or on the acidic. How about the trypsin? It works on basic. If you don't have enough basic condition, this trypsin will not work. And for the drugs, last things, we have two kinds of drugs or inhibition of enzyme. Some of them are reversible, some others are irreversible. It depends on the treatment policy. Mainly we prefer these reversible ones because like ibuprofen. You can see ibuprofen or gelofen? Ibuprofen. Yeah, it's a painkiller. It is a competitive and reversible. But for reversible, we have two kinds of inhibition. They are competing with the active site, they compete with the substrate, they sync to the substrate with the active site binding, inhibit enzyme. Let's say my work is that transferring to this side. If something looks like this and occupy my hand, what happens? It inhibits my activity. In competitive ones, the substrate same to drugs, something like this. I don't know, something, let's say we have something here, make my hands busy and I can't actually transfer this. It's a kind of competitive inhibitor. Many of drugs are like that. Many of drugs, like ibuprofen. But for the irreversible ones, how about the aspirin? Have you heard about aspirin? Yeah. What is that? Reduce the pain, chest pain. They work in the same enzyme, but ibuprofen and gelofen are reversible and competitive part of this reversible, but aspirin is irreversible. When it bonds to that, they destroy the enzyme. It takes 3 days to replace that enzyme. But for this one, after 8 hours, the enzyme works properly. Just reduce the activity of that in this form. There are some examples here, and one example that this is aspirin bounds to this specific place with ibuprofen and gelofen. This is example for antibiotic working. Antibiotic stops bacteria enzymes activity. So, and it helps as an antibiotic. And the final things, that's the final slide that we have. A general overview of factors influencing enzyme activity. Substrate. I am an enzyme. If I don't have a substrate, what happens? I can't work in high speed. I should have substrate to work. If I have more product, if I have, let's say this is my product here. If I have more product, this product inhibits my process. Else, don't work. That is enough. This is the enzyme pathways or metabolism pathway. Let's say when you have more glucose, when you need more energy, you consume that glucose to produce ATP. When you have enough ATP, what happens to enough extra glucoses? You don't produce ATP, you store them. Glucose is stored as a glycogen. When you have enough glycogen, what happens? Extra glucoses to go fats. So that is easy. Pathways depends on the substrate and product accumulation. If you have more product, it says please stop that process. And we have some hormones, they control these pathways. We are not going to study those ones, but you should know hormones control. The best hormone controls pathway, what's that? In diabetes? Insulin. Many of these enzymes are controlled by insulin. If you don't have insulin and you get substrate, if you have enough product, but that enzymes can't work properly, they should be controlled by hormones. Always remember, all of these enzymes are controlled by substrate, by products, and hormones. That's it. Thank you so much. I hope to see you again in the this?
دكتور